So today we are going to go over, honestly, one of the most difficult topics within our year of IB psychology to go over and understand and talk about, but also one of the most important in terms of understanding how the brain influences behavior, which is what we're doing in the biological level of analysis. So the first thing I want us to do is to watch this video from the Crash Course series, which we'll probably see more of this year and you may have seen in the past. This is going to cover a lot of topics, some of which are not necessarily going to be important to us as we move forward. But what I think this does is it does provide a good background of the neuron and how it sends messages throughout our body and how neurotransmitters specifically, the thing that we're most interested in for today, sort of send and change the perception of messages in the body and therefore influence behavior on the whole. So getting into notes, we really start off with the most basic cell unit of messaging through the body, and this is the neuron. And neural networks in the brain, so series of neurons in the brain, developed by the making and breaking of synaptic connections between neurons. And a synapse is really just a gap in between two neurons. And so these synaptic connections, and we'll see this in a second with the individual neuron, sort of reach out and make connections to different neurons, sort of setting up a channel through which messages can be sent. And the central nervous system really plays an essential role in coordinating this behavior. Neurons transmit sensory messages, so one's taken from the senses. So when you smell something, when you taste something, when you touch something, that sends messages through neurons to your brain to make sense of what those are, and then allow motor responses to occur almost instantaneously. So, for example, we talked about the hand on a hot stove. This involves our spinal reflex arc. And this is the simplest neural circuit. It tries to help us alive, or help keep us alive, I should say. So consisting of a sensory neuron and a motor neuron connected via a synapse in the spinal cord. And this allows us to make movement, and it travels at a top speed of 150 meters per second in sensory nerves and 0.1 meter per second in the brainstem. So a lot of it goes from, recognizing that something's wrong and sending that message back to the brain, and then slows down in the brainstem before allowing you to create the action quite quickly. So as the message is communicated from the brain and it passes from neuron to neuron, the message is carried across that tiny gap I just talked about, which is called the synapse. And the synapse is going to occur right in this area right here. And I'm sorry for my circle drawing. I'm still a novice at using the drawings on here. But as you can see, there's tiny little branches that jut out right here. And each of these tiny little branches can make connections with different neurons based on the ways in which your body has sort of made connections. And what carry these messages across from one neuron to the other are these chemicals called neurotransmitters. And like the title of the PowerPoint suggests, this is really what we're going to be talking about quite a bit, because these carry the impulse or message across to the dendrites of the next neuron. And based on the type of neurotransmitter and the type of chemical that is taking place, the message can be changed in a lot of ways. And the ways in which we process this message can be changed, therefore changing the behavior as well. So with neurotransmitters, there are many different neurotransmitters. And like I've said a couple times now, each type of neurotransmitter affects the behavior of the individual through the effect of the chemical itself. And this can include whether the impulse is going to continue traveling or not. It could amplify or reduce the effect of the message or impulse. And just as a general fact, the first neurotransmitter identified was acetylcholine in the 1930s, and that affects memory and a couple other processes as well. So a few different effects that neurotransmitters can sort of have an effect on. The first are excitation and inhibition. So as it says there, excitation is the trigger, they trigger the activation of the neuron. They sort of accelerate the car. And then inhibition tells the neuron to stop sending the message or reducing the activity of the neuron. But there's other substances that work to counteract the excitatory or inhibitory chemicals in the synapse or the neurotransmitters and clear them away after they've done their job. And these provide the basis for many different outcomes in behavior based on how well these chemicals are cleaned up in the synapse. The other types of effects that neurotransmitters can have on the traveling of the message in the neurons are agonists and antagonists. So these are types of neurotransmitters. And agonists increase neurotransmission activity. So these are chemicals that influence neurotransmission by imitating the action of neurotransmitters at the postsynaptic, so the beginning of the next neuron. They either block reuptake or increase their release. And so a chemical like Prozac works because they tell neurotransmitters to block receptors and therefore increase production of serotonin. And what this does for people taking Prozac, which is an antidepressant drug, so trying to stop depression from occurring, is that because serotonin sort of affects mood and low levels of serotonin have been connected to depression, the increased production of serotonin from the neurotransmitters being blocked and sort of flooding the system with more to get the message across allows the person to sort of be balanced out. The other type, antagonists here, decrease neurotransmission activity. So these are chemicals that influence neurotransmission by reducing the action of a neurotransmitter at the postsynaptic neuron. So for example, schizophrenic drugs help reduce dopamine production. For people who are schizophrenic, they have an overproduction of dopamine. And this helps to sort of balance out again or stop the production as much of dopamine. So a few different types of neurotransmitters that we have here. The first is acetylcholine, which we talked about just a little bit ago, but these are found throughout the brain and in neuromuscular synapses. They have a few functions: learning and memory, control of muscles, and they're really some of the most well-studied neurotransmitters that we have. The second one, dopamine. They include many locations, including the cerebellum, the basal ganglia, the limbic system, and they affect motor activity and coordination, emotion, and also memory. And then we have serotonin, which is an excitatory neurotransmitter. And it has many locations, including the thalamus, the brainstem, helps with sleep and arousal and mood, as well as sensory processing. Epinephrine is the last one there on this page. It employs and has a function dealing in emotion and stress and is included in the sympathetic nervous system. And just a couple others. The first one is GABA right here. So this is an inhibitory transmitter. It's found throughout the brain. It deals in anxiety, arousal, learning. And the last one, glutamate, found throughout the nervous system and dealing in anxiety and mood. So today we're really going to be focused on acetylcholine in terms of how we talk about these different studies. And the first one of these is Rosenzweig, Bennet, and Diamond from 1972. And the aim of this was to see the effect of the environment on physiological, processes in rats. And this was a convenient sampling. The rats were just available to them. It was a lab experiment. And what they wanted to do, and what they did, I should say, is that they had 16 total experiments done over 10 years. And the rats were randomly assigned to two groups for 4 to 10 weeks. One was an impoverished environment. It only had food and water. It had no toys, no sort of ways to enrich the brains of the rats. The second one was an enriched environment. They had 25 playthings and a new one given each day. So there was always newness. There was differences in their environment. And after they died, the rats died, slices of a brain sample were taken 1/10 of a milligram chemically, and it was analyzed and looked at under a microscope. And what they found was that in the enriched environment, the cerebral cortex, which deals in higher levels of thinking, more complicated processes, was heavier from the enriched environment than those that were in the empty environment, the lonely environment. And the number of neurons were the same. They just had larger neurons, showing a healthier neuron. They also had 50% larger synapses to allow for greater chemicals. There was also greater evidence of acetylcholine due to the appearance of this enzyme here, and glial cells which clean up for the neuron. So really what this showed was that the environment affects the brain through neuroplasticity. But more importantly for what we're talking about today with neurotransmitters, it also helped to increase the production of neurotransmitters as well. And because there was more going on in the environment, this helped the appearance of acetylcholine, which had an impact on their memory as well, because that's what acetylcholine does. So in terms of evaluating Rosenzweig, there is a reliability. It's been easily replicated. There is an internal validity within this. It does match up between what they're trying to study and what they carried out in terms of the experiment. It does show causal relationships. So this is quite a strong study in those ways. Other than animal studies and the limitations that come with that, a lot less limitations than you would normally see in a study. Building off of the Rosenzweig et al. Study is Antonova et al. From 2011. And this also was a study that looked at the role of acetylcholine in this time spatial memory for human beings. And so it's not an animal study, which makes it all the more better to establish a very clear causal relationship here between acetylcholine and memory, in this case spatial memory. So in the study, they had a sample of 20 healthy male adults. And you may be wanting to look at this because this is not a huge sample. So if you're looking at a possible limitation of this, it's the idea that a smaller sample may skew the results because you're not getting a full population of people. And the mean age was 28 years old, so no older individuals where spatial memory may have been affected uh, on terms that were bigger than acetylcholine. So they used a double-blind procedure where both the participant was blind and the researcher was blind at the time of the study. And the participants were randomly allocated to one of two conditions. So either they were put in a condition where they were injected with scopolamine, which was used as a beta blocker to block the transmission of acetylcholine, or they were injected with a placebo. And so the participants were then put into an fMRI, another good brain scan study, where they were scanned while playing the arena task. And this was a virtual reality game in which the researchers were observing how well the participants were able to create spatial memories. And so the goal was to sort of navigate around an arena with the goal of reaching a pole. And after they learned where the pole was located, the screen would then go blank for 30 seconds. And then suddenly, after 30 seconds, they would be put back in the arena, but at a completely different spot. And so their goal was to, as quickly as possible, use their spatial memory in order to get back to the pole. And then the procedure was repeated 3 to 4 weeks later, and each participant received the other treatment. So everyone in this repeated measures study was able to see the other study. And so what they found was that when participants were injected with scopolamine, they demonstrated a significant reduction in the activation of the hippocampus compared to when they received a placebo. And so what can we take from this? It again appears that acetylcholine could play a key role in the encoding of spatial memories in humans. So we're going to move into the next study here. And this is Caspi et al. From 2003. So in this one, they wanted to see the correlation between genetic type and responses on questionnaires about stressful life events and questionnaires diagnosing depression. And so within this one, we're going to be looking a little bit more at serotonin because this is a study on depression. And they took 847 white New Zealanders who were 26 years old. And what they did in this study is that participants were placed into one of three groups based on their genetic allele pair type. And they filled out a life history survey with questions about life events, so employment, health, relationships, those major things that oftentimes cause stress as well. And there was no difference found in the amount of stressors from participants of the three groups. At the time, 17% of surveyed qualified for a major depression diagnosis, and then the data was analyzed with correlations between depression symptoms, genotype, stressful events, and their interaction. So the results of this study were that participants with the two short alleles, that was just one of the groups, had a greater risk of depression after 4 or more stressful events over a period of 5 years. And so, when we're concluding that the gene type 5-HTT, or the gene 5-HTT, I should say, affects depression greatly, but only as it interacts with a stressful environment. The reason why we care about this in neurotransmitters, and when talking about neurotransmitters, is that really the fact that this stressful environment caused rates of depression, that points to lower serotonin levels and an imbalance of serotonin within the system. And so the fact that these stressful environments are affecting the body can in turn affect your behavior. if you're someone with depression, your behavior is naturally going to be changed by the appearance of this disorder. And so that is why we would care about this study within neurotransmitters. And it's also pointing to another fact that stressful events and stress in our lives can have such a negative impact on us. And we're going to continue seeing that as we go throughout this year. It's going to continue to point back to the effect that stress can have on us, not only just in the short term with the event itself, but long after the event has passed, going far into our future. A little bit before, we talked about agonists and antagonists, and when discussing the process of neurotransmission, biologists often refer to chemicals as agonists or antagonists, depending on what they do to the pre or postsynaptic receptor sites. So either the neuron it's coming from, the pre, or the post, which would be the neuron it's going to, the message. So all neurotransmitters are agonists for receptor sites. They are oftentimes referred to as endogenous agonists since they are biologically already part of our nervous system. So acetylcholine is an agonist for acetylcholine receptor sites. And drugs can also be agonists as well since they are external to our system. They are referred to as exogenous agonists. So they come from outside of us, or exo. So nicotine is an agonist for acetylcholine receptor sites, and short term it appears to have positive effects on memory. Long-term use of nicotine has a negative effect on memory. Antagonists, on the other hand, are drugs that block the receptor site and do not allow the neurotransmitter to do its job. They send an action potential down the neuron, which floods the system with more neurotransmitters of that type. So scopolamine is an agonist for, an antagonist for acetylcholine. Not all antagonists block receptor sites on the postsynaptic neuron. SSRIs, which are antidepressants and anxiety meds, they block receptor sites on the presynaptic neuron and prevent the reuptake of serotonin as a treatment for depression. As we talked about before, this causes greater production of the neurotransmitter serotonin. So the last study we're going to look at today is Rogers and Kesner, and this is from 2003. And this is another study that was looking at the role of acetylcholine. And just like the Antonova study, was also looking at the formation of spatial memory. So what they did was that they had 30 rats acclimate to a maze by placing food in one of the corners. And they allowed the rats to learn and become familiar with the maze and then get over being afraid of the environment before the experiment. So they are trying to control for the environment uh, and make the study more valid in that way. And then the rats were put into one of two conditions. Either they were injected with scopolamine or they were injected with the placebo. And this is very similar to that Antonova study that we just talked about. And then the encoding of memory was then assessed by the average number of errors made on the first 5 trials of day 1 compared to the last 5 trials of day 1. And then the retrieval of memory was then tracked by the number of errors in the first 5 trials of day 2 compared to the last 5 trials of day 1 as well. And so they're just operationalizing these different variables through doing that. So what they found was that those that were injected with scopolamine took longer, made more mistakes in the learning of the maze, and then the injections did not seem to have an impact on memories already created. And so maybe in this case, acetylcholine only impacts the creation of dreams and not actually retrieval of memory. And so acetylcholine may play an important role in the consolidation of spatial memories, is the key finding here. far as evaluating, the researchers did use a rigorously controlled experiment with a placebo condition in order to avoid the effect of confounding or extraneous variables. So this was a good part. Also, the study is able to establish a cause and effect, so it's a causal relationship. However, though, it's very much a reductionist or oversimplifying approach to understanding memory, because there are different types of memory. And so this could not speak for all memories possibly. It also the research could also one day lead to the development of treatments for people suffering from dementia or Alzheimer's disease. So this is an early study leading us possibly to, longer term solving for this dreaded disease. And then also biologists believe that animals can serve as models for human physiology and behavior, although this is simply correlative, right? And so the extent to which these findings can be generalized to humans is still in question and needs to be thought about when you're writing about this, possibly on an SAQ or something like that. The good news is that Rogers and Kesner is a example of both agonists and antagonists. And that's because acetylcholine is an agonist in the learning of the maze and the effects of acetylcholine on memory. But it's also an antagonist because scopolamine is trying to block the encoding of spatial memory. And so because of that, it is slowing down the neural transmission as well. All right, with that being said, that is the end of our notes on neurotransmission. Like I said, this is one of the hardest sections for students to understand year by year, but it is my hope that all of you, if you have questions, please come ask me, and please use all of the different resources on our class page.